All right. Sounds good. Thank you, Steve. Yeah, it is very slow. No worries, thank you.
Yeah, I think we can start a pseudo transect right now. So really just gonna try to go for time, or sorry, for distance. And I'll just step out as quick as I can see that we're safe. I'll just keep calling in the moves.
Looks like you're safe for 40 meters to
075 or
east. Yeah, you know me. I'm
yeah.
cardinal or intercardinal, man. That's as far as my little reptile brain can go.
Yeah. All right, you ready for it?
Yeah.
All right, so I think we'll have to wait till Argus starts to move before we officially start, but
Yeah.
we'll start from here. This feature right here, I think will give us some trouble. It's kind of large, so we'll see. Go around it.
That's an Audi, I think. No, looks like
No,
an Audi.
I think you're
Oh,
right.
it's an
It's
innie.
an innie. Yeah, so we will have to go trick the
Yeah,
eye.
so maybe your first...
Maybe we'll be all right.
what we could do, if you wanted, is we could drop back down to get just a little more shot in Argus.
Yeah.
Okay. Sorry, I was off comms there for a minute. I was talking to the boss about our manipulator. What am I doing here? I never got to look at the coral.
Well, better luck next time, Dan.
You can zoom out a little. Yeah, and we're a little dusty here. Sorry, I came in there in a it's emergency landing. It's going to cloud us up for a minute as I thrust down. Maybe I can do a car wash back and forth there. Oh, that's a lot of clay. I shouldn't have done that.
So, Rennie, let me know when we have started that transect.
Roger, yeah.
I got to clean us off here a little, so I'm going to back away from the cliff and thrust down. Mike, you can... we're going to want to come down, but... Yeah, I just want to come down and use Argus's sonar to, and if you could pan down. And we're going to light up the bathy there a little off to the east, see what we see. That will help us plan our ship moves.
Yeah.
Yeah, you can start coming down easy.
Just so the ship knows, the shoreside science SPL has been dropping in and out. So if you ask us a question and we don't get back to you immediately, we're trying to log back in.
That's all right.
All right.
Yeah, I'll keep track of that on the video side. I can see who's logged in and who's not, so
Cool.
I'll let the team know if you guys aren't on.
Thank you.
No problem.
We have at least 11 asking if there's any evidence of methane in the present location. We just jumped back on, but do you guys have an answer to that?
Any evidence of what?
We've been on...
Oh, sorry. It's not for me.
Methane seeps.
We were recently on one, right?
I haven't seen any since 11:30 when we came back on.
Since 11:30, you said? Okay, I'll tell Lisa we haven't seen any in some
we
time.
switched science at 11:30.
That's interesting.
So.
He's changed his heading a bunch.
Oh, come and look to the north. Yeah, Roger. Sorry, I'm using your sonar. Change your heading when we use sonar in it. Yeah. Yeah, so we're trying to get a feel of the how to plan our vessel moves here. All right, there's 40. Oh, you got 40 at least.
You have 40 at 060 or 075 maybe? 075.
Yeah, sure.
All right. Good
Yep.
for it?
Yep, good for it.
All right. Bridge nav.
Okay, Mike, you can change your heading as you will.
140 meters, bearing 075.
You can change your heading as you want. Let's come up a bit. This is where we were when we started, but you can come up quite 20 meters a minute there.
What speed do you want that at? Right now his transfer speed's set at a half a knot, so the
Sure.
first one will be quick.
Yeah.
And then it'll...
Okay. Yeah, we'll try half a knot. If that's too much, we'll slow it down.
Yeah, I think it'll probably be, you'll be going something like 0.3 and we'll just, by the time we sort out the next half a knot, move it.
Yeah, I'm good with that.
See what that
So that's a good depth there for us. If your guys in the back row and at home are happy with that.
Sounds good.
1085 is kind of our transect depth here for the moment. That puts Argus just above the wall of death there.
Once Argus starts swinging, we'll call it a start, but we'll hold here for now.
Copy.
And this will be start of the 8th transect this dive, correct?
Yes.
Yes, brilliant.
T8B.
Great.
That's what happens when you
Thomas
turn on auto heading against a cliff. Sorry.
Tom Leidig, who was keeping an eye on the last watch, has confirmed that there hasn't really been any signs of seep since that really big one at about 8 o'clock this morning.
What's that? Yeah, we're waiting for Argus to move there.
Oh, sheepdog sponge.
Yeah. A nice little glass sponge. We think we collected one of these earlier.
Leela, you can mark that as start if you haven't already.
Yeah, I did.
Okay.
Thank you.
Sheepdog sponge.
Yeah. Do we
Those
have
are
a scientific name for that, or is that, or no?
So this will be one of those. There's a big group of they're called lysocene.
Oh, the lysocene sponges. Okay, cool. So it's one of those. I think I remember how to spell that.
Yeah, LYSS.
I couldn't even pronounce it, much less spell it. Sheepdog I can deal with.
It's a good name for it.
Yes, this is a ship that named it Sheepdog, Abby.
Ah.
Yeah, I can't wait to tell my
Well,
parents I named a species. Or
I really think that should be the common name. I'm not gonna lie.
So we collected.
Oh, we're totally gonna use it.
Yeah. And there's a chance that when we were here in 2017, we collected a little barrel sponge that looks like the sheepdog sponge. And we're able to put it in the genus Hylascus. I'm not I mean, it's hard to tell just by looking at it and not looking at the spicules if these are the same, but it was also covered in these really long spicules on the outer surface. So I'm kind of hopeful that that might be this new Hylascus.
Cool.
Yeah. It's actually a genus that is apparently pretty common along the west coast.
Would you mind writing that one in the chat? I'm curious how that's spelled or saying it.
That's unconfirmed though. It's definitely a lysocene glass sponge, but
I won't write it. Don't worry. Just personally curious.
Mm-hmm.
Hyalaskis or Hyalakis?
Oh, I left out an S.
No worries. I got it.
Sorry.
All right, that ship move is complete. Argus still has about halfway to go.
Roger. Let's keep it moving if you want.
Yeah. Well, what I'm curious about is if we go around here.
Oh, there's a bedheader cone.
Yeah. What was that one, Rennie? A 20 or a 40?
That was 40, but
there's a curve coming.
We might, yeah, it might be a curve, but we could always drop down. You know, we'll still be in a 10, 20 meter window. If how much science? How much does the depth matter for this transect? Do you want to stay at a relatively constant depth?
Yep.
Depth doesn't matter so much as altitude. So I know it's a slope, but
yeah,
I think depth changes are fine.
Okay.
Wow. What are you?
Let's try another 20 on that trajectory. See what happens.
Yeah, yeah. We might drop down, but
I
that'll
think
be
that's some clavularia coral.
Wow.
That's
Great,
awesome.
And a Gersemia.
Swifty is sick.
Turn my gains down and goosed it there.
One more step. 20 meters. Was that?
I
065.
Can't remember those. Come down 5, Mike. Come down 4, please.
Tom Leidig, whose group will be analysing all of these ROV transects, is confirming that a depth change is fine. Tiny paragorgia.
Yeah, there are lots of little guys over here.
Yep.
Little corals, excuse.
Much smaller than what we saw first thing
those
this
were
morning.
ginormous. Whenever someone refers to a time of the day, morning, I don't know what day they're talking about or where in their watch schedule that falls.
That's what happens when you start your day at midnight.
Yeah, right.
Right. So Gersemia is on our collection list, so maybe when we finish this transect, we can find some more of that.
Sure.
And grab one.
What's that orangey coral to the very left of the screen now?
That's another one of those Swiftias.
Another Swiftia, okay.
Yeah, the fans. They're interesting because they have a red sort of body and skeleton, and yet the polyps are yellow. Yeah.
Oh, yellow.
So
cool.
it makes it look sort of orange.
Yeah, that's cool.
Looks like the next one's going to be more northish.
Concur?
Tell if I get around the...
Yeah, once we get around there, we'll be able to. Argus still hasn't moved yet from that last 20 meters. It should
Really?
catch up. But yeah, I agree. I think we'll need to tuck in there.
Yeah, I've come around quite a bit. I'm looking 315 now.
Yeah.
I'll square up to it here.
It's a cool rock face
Yeah.
canyon that we're going through.
Yeah, for sure.
Is that another really knobbly Paragorgia? They're so big.
And to answer Jenna's question, I haven't seen any signs of slope failures up to this point. There's some more of that Garsemia.
So which ones are the the Garsemia?
The purple, pinkish, poofy ones.
The lower ones.
Yes.
Gotcha.
So we had
start
floofy
moving them to the
Paragorgia,
north
and
now.
now we have
Ship's
poofy
done, right?
Garsemia.
Ship's done. Argus is still halfway.
Yeah.
But I think we'll do. I think we can probably do 40 north.
Yeah, sure.
Might cause some adjustments, but try it out. Bridge nav.
North or northeast.
40 meters north. Thank you. Yeah, I think we'll end up coming up here if the distance, distance over ground, it won't be a straight line or anything. So it'll just be.
Yeah, Raj. I've got the Herc at 5 meter divisions now. I think he's caught that, but just in case you didn't. It scans faster too.
Yeah, yeah. Yeah, it looks like we'll be able to tuck in there the other side of it. May pull you off a little, but we'll adjust.
And the north move should help me out.
Yeah,
yeah. Couple of sheepdogs and a thorny head.
Yeah, what's that crab tucked back in there?
Doesn't appear to be going north, though.
Not that we could tell from here.
He's going 315.
Because he's changing his heading at the same time.
So unident.
Like pirouette looking thing.
There's lots of these small sponges. Do you see that one in bottom left or bottom right, Abby?
Bottom right. Oh, like tucked underneath that little rock?
The little white one?
Yeah, I think
with a
they're
crab
just getting
next to
started. Yeah.
Sheepdog puppy.
So Abby,
I like it.
Abby, I had a question about how fast sponges grow. And I if we see a 10 centimeter sponge, do you know approximately how old it is? Like how long does it take to get to that size? I'm sure obviously it depends on different sponges, but is there a general guesstimate?
So there's been So generally we thought that they grew very, like most things kind of in the deep sea, very slowly. But and it's quite difficult to study growth rates on these sponges because they're quite hard to keep alive in sort of lab conditions. But they have done some experimental work in British Columbia, where sponges from fjords there. There's one species in particular, Rhabdocalyptus dorsoni, common boot sponge, and they actually found that it grew a couple of centimeters a year, which was kind of faster than people expected. And the other way that they can age them is by looking at shipwrecks and looking at the sizes of sponges on shipwrecks. So that gives you sort of a maximum age for that sponge. So
that's an interesting technique.
Yeah, that's cool.
How many centimeters a year did you say that was from that estimate?
I'll have to like double check to make sure, but I think it was kind of 1 to 2 centimeters a year.
So,
and that would be for one of those, like kind of a sponge that looks similar to the sheepdog sponges.
Okay.
I mean, considering they never see sunlight, that's pretty impressive, all things considered.
Yeah.
In this harsh environment.
Mm-hmm. I mean, they did. There's been more work done on kind of shallow tropical sponges. There's a paper on giant barrel sponges on coral reefs, and that came out. And they were sort of called the redwoods of the reef, because they think they estimate that some of those have been around for sort of thousands of years. That was, I think, that study was using growth curves and estimates based on observations of changes in sponge volume. But it is quite difficult because it's obviously not a linear rate. So there's kind of different ways to tackle that problem. Unfortunately, they don't have sort of handy growth rings like corals that you can kind of get at, because most of the time once a sponge dies, especially like those little the lysocene sponges, their skeleton just kind of falls apart. They become a little mat of spicules on the seabed, and that's it.
I think we can go another 40 north. What do you think? Kind of tuck into that canyon there.
It's a bigger primnoid.
40 north. Sorry, you're muted, Dan. I can't hear you. Okay, yeah, no worries.
Yeah, sure, sorry.
Yeah, no problem. Bridge snap. One more step, 40 meters north.
Let's do, if you do 315, that'll bring it closer to me. We're pretty far away.
Yeah.
And I'm moving north and he's moving north, so.
Yeah.
If you do 315, it'll bring it in closer. Or do a step.
Yeah, I'll do
step 10 to the west.
Yeah.
I'm gonna get pulled here shortly.
Right, yeah. Bridge snap. How much down do you have, or is it just sheer?
What's that?
How much down do you have, or is it pretty sheer? Like, could we get closer if we drop down a bit? But it looks like it's going to curve towards us actually a bit.
Meters.
So that north move, the cliff will come closer.
Yeah, but right now I'm at the end.
All right, so I can pause this and do a bump in.
Yeah.
Bridge now. Can I have, let's hold position here, and then I'll have one move 10 meters bearing west. Thank you.
Argus is out of my sonar range.
Yeah.
Is that a Faria, Abby, on the right-hand side?
Oh, yes.
But we're still in transect. Sorry.
We are. We're on transect, but yeah, that would be a great one to get a slurp of.
What were we looking at?
So this little lacy looking sponge in the center, just under the lasers right now.
Yep.
There's a few different species, and again in 2017 we collected some which turned out to be, so Farrier ocker is a fairly widespread species. It's one of the reef building sponges that you find on glass sponge reefs, just further north of here. But we identified one of the farriers collected near the sanctuary in 2017 as Farrier mexicana. It looks similar, but it has different spicules to Farrier ocker. In which case that's kind of a range expansion for that species. So we're keen that it's definitely on our collection list to have a look and see if we get if we can confirm that it is that presence of that different farrier.
Cool. Would you mind throwing that one in the chat too when you have a second?
Yes.
Thank you.
Is that some more of it, or is that something different coming through center right now?
Yeah, that looks similar.
It looks similar, but it does have a kind of an unusual morphology.
I think I can stack another 20 north on top of that. Argus hasn't moved west yet, though, but I think it'll still be good.
Yeah, it's moving west. It's moved 5 meters.
Has it?
Yeah, I'm watching it on the sonar.
Yeah.
It's coming in. But yeah, if you want to move to the north now, that'd be...
Okay. Bridge now.
Well, hold on.
Yeah.
It's to the north of me already. Let me get a little on the bank.
Yeah. Stand by, sorry. Yeah, I know the DVL keeps tricking me, but you're down here, I think.
Yeah, it's
yeah.
Nice on the cliff having it in sonar. I know exactly where it is.
Yeah.
Maybe I need a DVL reset. Oh, no DVL, that's why.
We could reset, we'll just lose it
altitude.
Again. Yeah.
Moving a little faster now.
There are a lot of single-stalked corals around here, or single-stalked morphologies of
Probably too
corals along this face.
I wonder if that has to do with the hardness of the substrate, and the big fans and the current would fall off.
Right, because of the angle of the substrate too.
Yeah.
Yeah, because usually in the current, I mean, they'll be kind of oriented in a way to take advantage of that, but it is also really steep here, so.
Yeah, and if it gets too heavy, it falls off.
Yeah.
That would be interesting to look along the bottom edge of the cliff just to see what's stacked up down there.
Right. Skeletons. Well, you were calling it the wall of death, right, Dan? So.
Yes.
He knew.
Yeah.
So I just had a quick fact check on the sponge growth rates, and they did find kind of linear growth rates of about 2 centimeters a year. those little rhabdicollectus dorsal eyes. So you could have like a little 10 centimeter sponge that's maybe about 5 years old.
Wow.
So that looks like a
Come up a little,
bamboo.
Michael, please.
I think I can do 40 north.
Yep,
This one right here, acidity on the bottom left.
Bridge, Nav.
Yes.
Yeah.
And we sampled one of those earlier.
We did.
40 meters
long
bearing
while
north.
earlier.
Thank you.
Some more Garsemia.
Little one.
Yeah, I'll slow down a bit.
No glass sponge.
Yeah, is that what that? What about the small white things in the upper frame?
I think that might be a solus little sea cucumber.
Oh, really? Up top? Well, it's gone now.
Yeah, I think so. Let's look out again.
Just curious, Nav, how far have we come along this transect?
I think about 80, 90 meters.
Okay,
60,
great.
And just about 100.
Awesome.
I don't know if you have the high pack view in channel 3.
Not up at the moment.
Okay, no, it's no worries. So it's just it's not a straight line, obviously. Just we're following the curvature of the canyon. So.
Nope, that's fine.
Deep sea soul, right? Is that deep sea?
Yeah. Beautiful.
I think they should make a wallpaper of
Yeah.
that pattern.
Deep sea soul wallpaper.
Mm.
Nav, are we about to put in a ship move, or do we have a second to reiterate all our names and who's on the line right now?
Oh yeah, we're good.
Okay, cool. Well, this is Leela on data. Nice to be back with you guys.
This is Marley on video.
Dan on Herc.
This is Renato on Nav.
The Fab Five.
Sure, who do we have with us over there?
So you got Abby on
we
side 2.
And Erica on side 1, and we just went over some clavularia that went out the bottom of the image, and here's some more that stick swiftia.
Ah, clavularia and stick swiftia.
And those little squat lobsters.
Oh, so those are squat lobsters, huh?
Yeah, with the long, long little front claws. Hopefully we'll start to recognize everyone's voices after a little while.
Yeah.
It's tricky.
Just in time to switch shifts.
Probably.
One of my favorite samples from the very first cruise I was on with Nautilus was this really cool pink spiny squat lobster from Hawaii, from the off the northwestern Hawaiian Islands. They're pretty cool looking.
A pink squat lobster. I didn't know there was such a thing.
Yeah, maybe later on we'll get a little zoom of one.
Try 20 at northeast. See what happens.
Yeah, it might stretch out by the time, but we'll just be quick about it.
Right.
20
northeast.
Bridge nav. .20 meters, bearing 045.
Yeah, got a little close there. Trying to stay 5 meters off the wall the whole time for constant altitude from the front of Herc.
The bigger thorny head.
That's in horizontal altitude.
So are these the short spine thorny heads? They're bigger than the others we were seeing, or is that still long spine?
No, the short spines do get bigger. One does appear to be a short spine.
All right, so 2 short spines and a long.
Falling behind, I need to speed up a little, I guess.
Especially with that new move. Back that way.
Yeah, looks like it's gonna hook around.
Yep. Matches the battery, which is good.
Is that a curled up little grenadier?
No, it's a hagfish.
That's a hagfish?
Yeah.
Tell by his beady eyes.
Yeah. I guess grenadiers don't really sit or like that.
There's purple in enemies.
Yeah. Shells of something.
I was wondering if some of those are brachiopods.
That's a soft octocoral right there.
Yeah, what's that on the
Is
bottom?
that the Umbellula?
Umbellula, that's it.
Yeah, that looks like Umbellula.
Sorry for the dust. I'm changing my heading a little rapidly here as the wall comes around.
It's a tiny sculpin. Out of shot.
Yeah, that's the one I'm not sure if we'll have leash for in there. That little bit. But we could always flip temporarily as we come around, and then he'll be able to again.
That's probably what we were seeing, whatever our last watch was. When we thought it might have been a stock crinoid, it was probably Umbellula, right?
Yeah, that's what I thought. I pulled up a picture of it, and I thought that's what it was, but couldn't remember the Umbellula name.
We can bump him 10 meters to the west. He went way up. Oh, that's the move you asked.
Yeah, I did a 045.
Yeah,
which would be pulling, because
There,
Argus is 270, so back that way.
you could bump him 10 meters west now.
Yep. I don't know about that actually, because that's
I'm
Argus
gonna
is,
run out.
oh, west, sorry, I
Yeah.
thought you said east.
Yeah.
Bridge now. 10 meters west. Got my left and right mixed up.
I do that all the time. So we should have gone north there. I think he went more.
Well, yeah, it's an unknown box canyon, so it's hard to get.
So Abby, does Berea fall into the glass sponge realm?
Yes.
Okay.
Yeah, it's different from the lysocene glass sponges. It's one of the, when you look at it under a microscope, it's got a spun. Its glass skeleton is made up of fused spicules. It's like a fused framework. So
Come down 5 if you want, Mike.
Unlike the other little, like the sheepdog, for instance, when a farrier dies, its skeleton remains
intact.
Should get
some relief here shortly. The ship moves.
Do a lot of hexactinelids have fused spicule skeletons? I thought I had heard that.
I think it's a minority that have that.
Oh,
There's
okay.
yeah.
Wonder what I'm thinking of then.
Well, there's definitely, so there's only 3 species that are known to form sort of sponge reefs, and that's the Heterochronicalix, Barrier ocker, and Africalystes vastus.
Yeah,
I
it's an
think.
octopus. See him hiding down there?
Yeah.
Octopus at the bottom of the screen.
Yeah, still on transect though.
Maybe what I was thinking, do they have syncytial cell structure, syncytial tissues, glass sponges?
Deep water.
Sorry, I muted myself. So unlike demo sponges where they have kind of individual cells, they do have these synctal tissues, which is almost like, yeah. They're quite different on a cellular level to the demo sponges, which dominate shallow water sponges.
Okay, maybe that's what I was thinking.
There's a researcher called Sally Lees, and she has done a lot of work on the physiology of glass sponges, and even has some amazing footage of sponges creating their spicule scaffolds and maneuvering spicules into position during their kind of growth and tissue regeneration. So the glass sponges all have these, as you said, they're called the hexactinelids, named after these particular 6-pronged spicules that they have called hexacts. And that's something that all the glass sponges have in common.
Well, big anemones.
USBL and DVL now agree, so I'm not asking questions. I didn't do a reset at all, ever.
Yeah, they do look closer now.
Bit strange.
several liponema there and some long spine thorny heads.
Liponema, was that the anemone or?
The pom-pom anemones, yes. Little tiny thorny head. Right.
Yeah, cutie.
We have lasers right now.
Yeah, what's happening there? Too close. Sorry.
What kind of anemones on the right, about to come into the right frame? We had been seeing a lot of those in the past as well.
We're good for another 10 at
The,
045.
Those I don't know. They could be a Paractinostola. There are several deep
Bridge
water
nav.
species that are hard to tell by imagery without having it
Could do
actually
20
in the
if you
hand.
want, if that makes him happier up there.
But we do see a lot of Paractinostola
Yeah,
from these deep toes on the trawl survey.
Each ring in the sonar, is that, how many meters is that?
On Argus on the right is 20 and Herc on the left is 5, but we adjust those, but normally it's around there, or 10 and 20 or 5 and...
Each ring is 20?
For Argus on the right, yes, 20 meters.
Pacific
And it's
Grenadier
not
cruising by.
it's not north referenced, so that's vehicle forward is up.
Gotcha.
Yeah.
We so need a compass input. Hey, maybe that's a weather project.
Yeah. How can we remember that? Where do you, does his notes go somewhere better?
What's that?
Do his notes go somewhere that
No, they
they'll
go.
read them?
They go.
Because
No one looks at them.
that's the same.
They sit on the workbench for weeks, and then I finally bin them. I file them in around.
That's a good idea, though, to see if we can type that in.
It would make the moves a bit more intuitive.
Yeah.
No, the sonars will accept an NMEA string or the heading.
Oh.
And then there's a compass rose on the outside of the range rings that moves with the vehicle headings. So if you're looking at something, you can work out which way to move it without doing the mental mind game that we have to do here.
Yeah.
Rennie, how far have we gone in the transect now?
Roughly 160 meters.
How far are we trying to go?
200.
200, cool.
200.
Bridge nav.
So what I was asking about earlier, the little white
10 meters,
I
045.
don't know what they are. It's kind of below the lasers right now. What is that?
Yes, that's a little sessile sea cucumber.
Really? Okay.
So let's see its little tentacles out feeding. Which grenadiers are we seeing here, Erica?
I think most of them are Pacifics.
Pacific Grenadier?
Yes, that one might be something different. It's a lighter color, and its face and nose structure looks a bit different. So potentially we could see Popeye Grenadier or definitely Giant Grenadier. I think we saw some of those earlier this morning
There's
on
a little
previous shifts.
squatty in the hole. It's not pink though.
Oh, there they are.
Two of them.
Hiding behind a sponge.
What the? Turn the gain up on that. Set. It should be lighting up.
Wall.
Yeah.
There's so many squat lobsters everywhere on these canyon walls.
Yeah, it's like, what do you call a herd of squat lobsters? Flock?
Yeah, what's
School?
the collective noun for that? We gotta come up with one, guys.
Might need to come up the tether in the camera over there.
Yeah, it's
Okay.
I've been watching it. The bounces, get it?
Yeah.
Apparently the collective noun for crabs is a cast.
A what?
A cast. Cast of crabs.
Cast. Interesting.
Think I can go 20 at 90?
Where am I?
You're north of
Yeah, sure. Yeah.
bridge nav.
We got sheepdog sponge, center stream right now.
Yeah, we've been seeing those again now that we're back on this
You can hold that and we'll do 20 meters, 090.
And a little, possibly a little patch of farrier as well.
Thank you.
Come up a little, Mike.
A
Come
faria?
up 5.
I think
Right there,
so,
just
yeah.
going off the left-hand
bottom
side.
left, yeah.
Once we're off transect, that'd be something to look out for. If we happen to find farrier that's near one of the sheepdog sponges too, it'd be nice to get just a snip of one of the sheepdogs, just to confirm that it's the same as we collected earlier. There's an unfortunate tendency when you collect these little glass sponges that you think are the same based on what they look like, they turn out to be something different once you get them under the microscope.
Well, Abby, I can say that they don't snip.
Yeah, that's gonna be a grab.
Oh, they don't snip.
Okay. We can grab.
Yeah,
we can grab a snip.
We can snip with the. We can snip with
they just
the
don't
nip.
slurp.
They didn't slurp.
Yeah,
that's for
we
sure.
tried to slurp.
That's the one.
Yeah, we don't have to collect a whole thing if you do not want to.
No, it was great. I mean, you got that. I was up for that first sample. It's fantastic. And this will just be to confirm that it's the same spicules present.
Yeah. Well, I will make sure not to put it in the same box then.
Oh, we can
Yes.
Not
put
that I
some
would ever do that
spicules
with sponges.
we can put some spicules in number 1, and then you can compare. You know, they have different ones in there or same.
No, it's hard enough.
Well, yeah, because then what if both of them have both of them, Dan? Then what?
I don't know.
Oh.
Punt.
No, the dream is you get something that's easily identifiable by morphology, and that means that we can be confident when we're doing kind of the ROV surveys or surveys with the AUV that we can consistently name things and be confident. Or if we find out it's not, then just consistently lump them as well, and you know you've got a species complex, and that's kind of as fine scale a resolution as you're going to get. But
gotcha.
Yeah.
Crinoid.
Looks like a plexurid coral there.
Oh yeah, is that plexuridity?
A small sponge.
Oh, there's one that didn't quite hang on.
Several plexurids on the screen right now.
Mm-hmm.
Another Chiomycetes tanner crab there.
Oh, it's tanner crab. Okay.
And I think he is unimpressed with the ROV.
For another bump, I think.
I think that's actually the end of the survey.
Is it?
Yeah, the last Argus move will be, so just a little bit further for Herc. And
Roger.
All right. So we have a request from Tom in the science chat that if we see any knocked over sponges or corals, to have a little look at the base and see if we can identify if it broke off.